Hello, I welcome you all to today's session where we are going to discuss the next set of biomolecules, that is the proteins. The term protein is derived from a Greek word which is known as proteos, and that means holding the fast place. This is the basic word from which protein is derived. It was Berzelius, which was a Swedish chemist, who had suggested the name proteins to the group of organic compounds that are most important to life. The protein term or the specific word was used by the Dutch chemist Mulder. So here we have got two names, that is Berzelius as well as Mulder, who are credited for coining the word protein. And proteins are defined as, it is actually Mulder's definition that we are studying nowadays, which defines that proteins are high molecular weight, nitrogen rich compounds which are the most abundant substances present in animals and plants. So basically we have got two terms, that is they are nitrogen rich and have quite a high molecular weight. Proteins occupy a central position in the architecture and functioning of the living matter, and you can classify proteins broadly into two categories. So they can be classified as the structural proteins and the dynamic proteins. One category is structural, the other category is dynamic. So let us come to the structural proteins. So structural proteins are essentially the proteins which are responsible definitely for maintaining the structure and strength of the body. You remember the term collagen, elastin, which are a part of the connective tissues, or bones or cartilages, which are present in their matrix. Sometimes they are present in blood also. So they are one of the major category of structural proteins. Another we can also think about, that is the keratin. That is a major component of your epidermal tissues like your skin and hair. So these terms you are quite familiar with. Now let us move over to dynamic proteins. As the name indicates, they must be performing various different functions of the body, and because they perform dynamic functions, that is why they are said to be dynamic proteins. They mostly include the enzymes, they will be including hormones, they will be also including the blood clotting factors that we have discussed in our chapter on blood vascular system. They must be including the defense proteins, that is the antibodies, which are scientifically or in medical terms known as immunoglobulins. They must be including the membrane receptor proteins, which we have just now completed in our chapter on chemical control and coordination, that is that of hormones. Then they must be including the storage proteins. This group must also include the proteins which are involved in gene regulation that we will study in higher classes. It also includes under its ambit the most important one. How can we forget this? That is the hemoglobin, the oxygen or the carbon dioxide carrier that is present in our blood, as well as the proteins that we have studied in our chapter on muscle contraction, that actin, myosin, troponin, tropomyosin, these all proteins will come under this category. Basically, dynamic proteins perform various dynamic functions, and they are popularly called as the working horses of the cell, because they carry out most of the vital functions of your body. Then let us come to the elemental composition of proteins. What are they made up of? So basically, proteins are mainly constitute, or they, I should not say constitute, mainly they are made up of five different things. One must be carbon. It is present in the highest proportion in the protein, making up 50 to 55% of a total protein, followed by oxygen, which makes up around 19 to 24% approximately. Next predominant element is that of nitrogen, because in the beginning I told they are nitrogen rich compounds. So nitrogen makes around 13 to 19% of the total protein. Plus it also consists of hydrogen, that is around 6 to 8%. In some books it is mentioned 7.3% also. And the last element is sulfur, because in not in all amino acids sulfur is present, so that is less than 1% of the total protein. Besides the above, proteins may also contain other elements such as phosphorus, iron, copper, iodine, manganese, magnesium, zinc, etc., depending upon their functionalities, particularly when they work as enzymes. The content of nitrogen is an essential component of proteins, as I am telling you repeatedly and many times. And estimation of nitrogen in the laboratory is also used to find out the amount of protein in body fluids and foods. And there is a method which is called as Geldel's method, which will help you to determine this. Then let us come to the next part, that is how proteins are considered to be polymers of amino acids, means they are chains of amino acids. So under this heading, we are supposed to understand it is the amino acids which are considered to be the building blocks of proteins. Means like a wall is made up of bricks, chains of proteins are made up of individual units or monomers which are called as amino acids. Amino acids are most probably the first molecules that were present in the atmosphere of Earth, or they were formed in the atmosphere of Earth. When we will study the chapter on evolution, then you will come to know about it. The function and the shape of the protein is, you will be surprised, it is affected by 20 different amino acids only. So whatever proteins that you are able to encounter in the biological system, essentially they are made up of only 20 different type of amino acids and two amides. Of course nowadays we are getting the 21st and 22nd amino acid, but they are not very predominant. So still the popular opinion goes that whatever proteins are present in our body, they are made up of basically 20 amino acids and two amides. When I am repeatedly telling that they are amino acids, so basically in their structure, they must be containing an amino or NH2 group to justify this part of their name, and an carboxylic acid group to justify this part of their name. So basically in an amino acid, there will be the presence of a NH2 and a COOH group. And if I write the structure of the simplest amino acid, it will be having a carbon at the center, and the four valences will be substituted by the hydrogen atoms as well as the carboxylic acid groups, including the amino group also. So here we will find that the hydrogen atom each are attached to the carbon which is located next to the carboxylic acid group, as you can see in the structure, And this here I have written that it is H group, but we can write it as the R group, because you will find that this part of the structure of amino acid will remain same in all the 20 different amino acids, only this group will be variable. As when I was writing the structure, I told that I am writing it for the simplest amino acid, so that's why I have substituted this R group by a hydrogen atom. As we discuss the different type of amino acid, you will come to know what I am talking about. So having said so, let us discuss that what are the different type of amino acids. So the principal amino acids can be classified as follows. The first one is the neutral amino acid, and the examples under this category will be glycine, alanine, valine, leucine, isoleucine, etc. So neutral amino acids are basically the amino acids which have one carboxylic group in their structure, as well as they have one carboxylic acid group and one amino group in their ring structure. So if I go on writing their structure, let me write it for alanine. So for this one, if I am going to write it, then the structure will be, this part I have already told that it will be remaining the same. And this R part I am substituting by the CH2 group. So that makes alanine. The second group, that is called as the acidic amino acid. So definitely the name indicates this COH group, that will be more. So they have an extra carboxylic group. So they are otherwise called as monoamino dicarboxylic acids. And the example under this will be aspartic acid, asparagine, glutamic acid, glutamine, etc. So aspartic acid, glutamic acid, their basic structures like asparagine, glutamine will also come under this category. So let me write the structure of aspartic acid, and the structure will appear somehow like this. Let me change the orientation a little. Let me write the carboxylic acid group here, the amino group over here. Here is the H group, and let me write the functional group over here. So it should be a CH2 and another COH. So here you find one COH, here you find another COH and one NH2. So that is why they are monoamino dicarboxylic. Now let us move over to the next category, that is obviously the basic amino acid. As we have written the acidic amino acid previously, so the next category will be that of the basic amino acid. So obviously they will be called as diamino monocarboxylic acids. Diamino means with two NH2 group and one carboxylic acid group. And the popular examples under this category will be those which are present in histones, that is arginine, lysine. These kind of amino acids will come under this category. If I try to write their structure, then it should be something like this. Let me change the orientation so that it is easy for you to understand. This part, it almost remains the same. Now you see how the other chain is there. There will be three CH2 groups in chain like this, and the fourth CH2 group is attached to another amino group. So here is one amino group, here is one amino group, diamino monocarboxylic acids. I hope I had made this clear. Then the next category is the sulfur containing amino acids. And these are one of the rare types. The sulfur containing amino acids have the examples cysteine and methionine. This methionine we will come across in our genetic code more often. So if I try to write the structure of cysteine, so somewhere I have to give a sulfur group to it. So let me write the basic structure once again. Up to this it will remain the same, and the sulfur containing group will be present like this. This is the sulfur containing group, so that is why it is called as cysteine. But because to accommodate this you can say steric form, actually the orientation is H over here, NH2 over here, CH2, which is attached to a sulfur group. So this is the actual configuration. Then the next category will be alcoholic amino acids. Name indicates they must be having the presence of alcoholic or hydroxyl group in them. So serine, threonine, and if I am remembering it correctly, tyrosine, then they are under this category. Serine, tyrosine, these are the members under this group. So if I write the structure of serine, then basically let me write the structure normally up to here. Up to this it is remaining common, as you can mark. So here is a CH2 group, and to this C, OH is attached. So because of the presence of this, it is called as the alcoholic amino acid, serine. Structure I have written. The sixth category, that will be aromatic amino acid. So obviously they must be having a cyclic structure with a straight side chain, having the carboxylic as well as the amino group. So the most common example under this will be that of phenylalanine. And phenylalanine will be having a structure, if I write it in most of the books, the ring is given in detailed form, but I will not write the ring in details. This is the aromatic ring. It is having a CH2 side chain, which is again attached to a CH2 NH2 group, and here is the carboxylic acid group. So this is the simple structure possible for phenylalanine. In some books, this particular ring you might get in more details. The last category is that of heterocyclic amino acids. So as the name indicates, they have nitrogen in their ring structure. So here the example will be histidine, proline, tryptophan, etc. Okay. So now come to the next part, that the 20 different amino acids which we have discussed, and the 2 amides, they are mostly present in proteins, so they are called as the protein amino acids. And except glycine, the alpha carbon is asymmetric, and the amino acids are generally levorotatory, and certain non-protein amino acids are dextrorotatory. The concept is exactly opposite to that of the carbohydrates that we have studied. So let us understand how these protein sequences or the chains of amino acids are determined. The incorporation of amino acids one after another is controlled by triplet codes. Once you read molecular basis of inheritance in class 12, that will be more clear to you. But please understand, for the time being, whatever is the linear sequence of amino acids that are present in your body, that is already pre-decided in the triplet codon form, and it is encoded over your DNA. So when DNA gets transcribed into the messenger RNA, the sequence of amino acids to be synthesized are passed on to the messenger RNA as such. Because protein synthesis takes place in cytoplasm, messenger RNA comes out of the nucleus, and in the cytoplasm, with the help of the cellular machinery like ribosomes, endoplasmic reticulum, ATP, various enzymes, calcium divalent ions, magnesium divalent ions, it synthesizes the proteins. So incorporation of protein into amino acids, we can be saying that it is indirectly decided by the DNA. A protein can also have non-coded amino acids, means there are some amino acids which are essentially called as the rare amino acids. So these rare amino acids won't be having these rare amino acids won't be having any genetic code, means they are not encoded over the DNA. They are derived from the coded ones through various modifications. Like proline will be coded by DNA, but if I am making hydroxyproline out of it, so that means I am doing some modifications to proline. So hydroxyproline is a rare amino acid. Similarly, from lysine I can make hydroxylysine. Both of these rare proteins are found in your collagen tissues. Then next, let us come to the category that are otherwise called as the non-protein amino acids. Means these amino acids are present in your body, but they do not form proteins. Remember urea cycle. So they are many in number. And firstly, I can say they are present in the free state as well as they can be present in the combined state. Some of them are present in the antibiotics. They are called as the amino acid analogs. And the second category is obviously that we find or encounter in various biosynthetic pathways like urea cycle. Remember ornithine and citrulline that I have spoken to when I was teaching you urea cycle. These are the non-protein amino acids. Homoarginine and diaminobutyric acid are also a source of nitrogen. And another important form, you will laugh by its name, this is called as GABA. And the full form is gamma aminobutyric acid. And it is an inhibitory neurotransmitter, acts exactly opposite to that of acetylcholine in your brain and some other parts. Another example will be diaminopimelic acid, which is an intermediate in the pathway where lysine is actually synthesized. So this is one classification that I have told. Another classification that you will encounter in some of your textbooks, that will be into grouping the amino acids into three categories as follows. They will be essential, non-essential and semi-essential categories. So let us discuss what are they. The essential amino acids are the amino acids which cannot be, means your body cannot synthesize them. So that means if they are not produced in your body, then you have to obtain it from outside. So from outside, how you are supposed to obtain it? You are supposed to obtain it through the food material that you take. Particularly they are present in plants. Valine, leucine, isoleucine, phenylalanine, lysine, tryptophan, they come under this category. These are not the amino acids that your body synthesizes. So that means if your diet is lacking in these amino acids, then you will be facing serious problems. Non-essential amino acids can be synthesized by a particular organism and it may not be strictly required in your diet. Serine, cysteine, if I remember properly, proline, glycine, tyrosine, they come under this category. And the semi-essential amino acids, they are particularly required by children of your age who are in their teens or growing age, and particularly the mothers who are actively breastfeeding their baby. Particularly histidine and arginine will come under this category. Now having said and read this, now let us come to the 21st and 22nd amino acid that I have talked about. 21st one is selenocysteine. The 20 amino acids already I have told you, but in recent years we have discovered a 21st amino acid known by the name of selenocysteine, and it is found as to be present in the active site of certain enzymes or proteins. For example, glycine reductase, then glutathione peroxidase. In these particular enzymes on their active sites, this amino acid selenocysteine is present. It is an unusual amino acid because it contains selenium in place of sulfur. Till now I have told that there are so many different categories and nowhere I have talked about selenium, but it contains selenium. And incorporation of selenocysteine into the proteins during the translation is generally carried out by a stop codon. Generally, stop codons, we have the idea that whenever they are present, they stop or they terminate the protein chain. But there is a stop codon, as you will study in class 12, that is called as UGA. And this particular one is generally responsible for coding selenocysteine. How it is possible, still the area is under active research. And I have told you about a 22nd amino acid also. The 22nd amino acid is otherwise called as pyrrolysine. And in the year 2002, actually, some researchers have described it, and still the stop codon UAG, last one I told, this is coded by the stop codon UGA, and this is coded by the stop codon that is UAG. But the exact functioning and their biosynthesis is still under active research in recent years. Exactly how do they work, how they control the metabolic pathways at all, we are still carrying research in that. Having said this, now let us understand how there is the formation of peptide bond and how amino acids, they work as or they function as dipolar ions. This is very, very important, that how these amino acids, they function as dipolar or Jupiter ions. Amino acids in a solution which is at neutral pH, they exist predominantly in this Jupiter ion form. In this dipolar or the, you can say, Zwitterian form, the amino group that is protonated, means instead of being present as NH2, this group will be present as NH3 plus. And similarly, the carboxylic acid group is deprotonated. Instead of being present as carboxylic acid, it will be present as COO minus. And the ionization state of the amino acid will vary with its pH. That means in acid solutions, if it is having a pH of 1, the amino group will be protonated and the carboxylic group will be not dissociated. Means at pH 1, it will be NH3 plus, but COOH on the other side. And as the pH you gradually go on increasing, the carboxylic acid group is the first group which gives up the proton. The dipolar form persists until the pH approaches 9, and then the protonated amino group loses a proton. And at the pH at which the amino acid is electrically neutral or the molecule exists as a deuterion, that is called as the isoelectric pH. And it is the middle pH between the pKa values of either side of the isoelectric species. So that you will learn in your chemistry classes that what exactly this means. And basically they will be present in this particular isoelectric point if they have to, you can say, function in a proper way. So generally you will find that the acidic or the basic proteins at a physiological pH of around 7.4, which is the pH of most of our body, the protein may be having more negative charges, and such proteins, they will be rich in basic amino acids like lysine and arginine. Generally they are found in histones. Acidic proteins will be more rich in acidic amino acid, and they will be the you can say near 7.4 pH, they will be existing with their isoelectric point. And I think the concept of dipolar and the Deuterion form is clear to you. Now let us study what is all about the peptide bond formation. So essentially the peptide bond formation tells you that how we are going to join two successive amino acids. So suppose I am writing the first amino acid like this. This is the variable group, let me write it as R1. And this is the second amino acid, let me write it in this way. Now this OH and this H, they will undergo dehydration to remove a molecule of water. Now there will be bond formation between this carbon group and this nitrogen group. So it will result into a structure which will be essentially, now see the peptide bond. This one is the peptide bond. Now here is the remaining H of it, because one H it is already giving up as the water molecule. The rest of the parts, I don't think there will be a problem for you to understand. So the part which I have highlighted, this part, this is the peptide bond. So basically, if I want to say, amino acids are linked serially by the peptide bonds, and this is the bond formed between the amino group of one amino acid and the carboxylic group of the adjacent one. In this reaction, a molecule of water is eliminated, that is dehydration process taking place. The chain containing the two amino acids linked by a peptide bond obviously will be called as a dipeptide, and there are three amino acids if they are linked in a linear manner, then it is tripeptide, and if they are numerous, then it is a polypeptide. The sequence of amino acids present in a polypeptide is specific for a particular protein. However, a single polypeptide may be at least 50 amino acids long, so that you can call it as a protein. So let us end our discussion over here. In the next one, we will be taking up some more aspects of protein structure. Thank you.